this in this chapter and in the next chapter. Okay? And what did we say about development was really cellular growth, differentiation, and one other thing. Morphogenesis. When we said what was morphogenesis? A change in shape or structure. All right? And part of this, related to that change in shape and structure, is the process of apoptosis. All right? Or, depending upon how you pronounce it, some people pronounce it apoptosis, whatever. Apoptosis, sometimes the P silent, it doesn't matter. Same thing. Okay? Although a lot of cells are growing, and cell growth is a major part, probably the biggest part of development, right? Going from one cell to 2 to 4 and so on, all the way up to the billion cells you have. We have to produce more cells, right? However, at some point in some areas, there are cells that are destined not to grow anymore. They have to die. All right? Example. Tadpoles. What do they look like? Here's a little frog and a tail. Some legs, right? Tadpole. Okay? What does a frog look like? Does it have a tail? No. Here's a frog, right? Eyes, no, little leg, feet, right in there. There's a frog. Okay? My awful drawing of a frog. But it has no tail. What happens to the tail? It shrinks. Those cells of the tail die off. They are genetically programmed to die. Okay? So, apoptosis is best described as what we call programmed cell death. The cell is going to die, but in a very ordered fashion. It's like we want to take down a skyscraper in the middle of downtown Springfield. You have a very ordered implosion so that all the debris doesn't go all over the place, right? So engineers go in, demolition engineers go in, and they pick out, you're going to break down this one, that one, right? You have a bunch of dynamite go off, they have this part, and then everything kind of folds in on itself, right? That is a program of demolition. A similar thing happens in the cell. There's a program of cell death, and it happens in ordered fashion in order not to influence the other cells around it. All right? This also occurs in cells that are infected, damaged, or at the end of their life. All right? So cells damaged, you can stimulate and start this program so that the cell dies, so it doesn't go on, right, and propagate its problems, mutations, all right, or abnormalities. What does this impart? So this is an example of programmed cell death. This is a cell membrane, right? Cell, nice round cell, happy, healthy, whatever it might be. The one next to it is what we call blebbed. All right? There's a disruption in how the membrane is formed. All right? Parts of the membrane aren't formed correctly. The cell is abnormal. Okay? Within that, there's a whole bunch of signal transduction pathways that are ongoing and causing this to happen. The cell is basically degrading from the inside out. All right? The DNA is being broken down. The proteins are being broken down without causing problems to the cells next to it, though. All right? So during apoptosis, DNA is broken up and organelles and other cytoplasmic components are fragmented. The cell becomes multi-lobed, or what we call the blebbing appearance. Right? And then the vesicles are engulfed by other scavenger cells. What it does, it protects neighboring cells from the damage that is inside. All right, so break it down internally so it doesn't leak over to others. This is essential, all right, for the development and maintenance of all animals. All right, development. you'll see that this is important in a lot of different aspects. A tail regressing is one of them. Okay? Yes, at one point you all had tails as embryos. All right? And they regress. Okay? And they have a remnant that's left there, which is your tailbone. Right? Leftover coccygeal vertebrae. Okay? Those are still there. All right? But the nervous system, morphogenesis, the production of the nervous system, how the brain is formed in neurons, is apoptosis, dying, or neurons dying someplace and being reformed, or cells dying someplace and being moved, and other functions being taken up other places. Very important. The fact that you have fingers and toes, right? That morphogenesis is dependent upon apoptosis. Because at one point, the limb grows as what we call a limb bud. It grows like this. Looks like a paddle. Okay? Eventually, the cells will start to accumulate in the area of digits. Right? And the area in between the digits is still cells. But eventually, the area between the digits does undergo apoptosis, leaving your 5 fingers and toes, in which there's typically no skin between them, right? Some cases there are. Some people may have webbed feet or webbed fingers, all right? There's also a possibility of polydactyly, all right? Are like this is what it looks like, okay? Here's your interdigital tissue. The cells undergo apoptosis, leaving a space between digits 1, 2, 3, 4, 5. Okay? So the fact that you have fingers and toes and they are separate from one another is a result of apoptosis of the cells within that interdigital tissue. Okay? So it is an important process. It is not a simple process. All right? We are overviewing it and giving you an introduction to it, but it is a multifaceted, complex process, which has several different starting points, one end point. Okay? And there's a host of different pathways that are involved in this process itself. Okay? Questions on morphogenesis? All right? So we're talking about development. I've given you the process of apoptosis and relating it to its importance in morphogenesis. All right? The next thing we're going to talk about is pattern formation. We go back to that frog video I showed, okay, where the cells begin to divide, 2, 4, 8, and then they have a pattern of dividing, right? First one is 1 into 2 down one plane, and then the division is perpendicular to the next plane, and then there's one that's below, right, and then so on and so forth. That is a pattern. That does not just happen in one frog embryo, it happens in all of them, in a process, all right? And the pattern formation in all embryos, all development, is important for determining front to back, top to bottom, right to left. Okay? Because insects have a distinct head and a distinct back. Legs are someplace where wings are not. Okay? This is important for pattern formation. Right? So it's a spatial organization of tissues and organs, where things are. All right? This is very important because where there's problems with that, you end up, like, say, in Drosophila with legs on your head and wings on the head and not on the thorax or on the abdomen, and problems. All right? So this positional information is dependent upon molecular cues. All right? That control pattern formation. And we're going to talk about one which is very important. We're going to talk about one called bicoid. It is a transcription factor that determines where the front or the head is in a fly versus where the back is. Okay, so we're going to be able to move things around and based upon where these proteins or mRNA or genes are expressed, where there's front and back, left to right, top to bottom. All right, so this will determine body. We use Drosophila because it's a good genetic organism, lots of generations that can be done, and over time you can combine your anatomical, genetic, and biochemical approaches. You've discovered basically that there are principles found in Drosophila that are shared and conserved across animals, even all the way up to humans. Okay? So, because at one point, even humans were just a big ball of cells. But at some point, we had to know what was top, what was bottom, what was in, what was out. Okay? And we go back to I'm going to keep referencing the frog video again. When cells started moving, the cells started growing, there were a bunch of gray cells up top, right? And there were a few yellow cells underneath. So it kind of looked like this, right? One top, one bottom. And then all of a sudden, you saw those gray cells moved and then left a little, kind of looked like a little lip like this, right? Once those cells moved, now they're under a different relative pattern. All right, now there were some cells inside versus what was outside. All right, that's what's called the blastopore lip. That ended up becoming the anus of the frog. So be it. But we're gonna use Drosophila as an example. So we're gonna talk about the life cycle of fruit flies. They're relatively quick, okay? And we're gonna build on some of our vocabulary and some of our knowledge from before. All right. Drosophila embryos look like this. They have this distinct front side, kind of like that, and they have different segments. They're segmented. And basically they're numbered 1 through 13. Okay? So there's 13 segments, segment 1 being the front, segment 13 being the back. Okay? So it's an ordered set of segments. All right? And you'll see what this looks like in a second. But the cytoplasmic determinants of the egg actually determine where those segments are. Okay? So this is a fertilized larva of a fruit fly. But the egg will also look something similar. It'll look something like this. Right? It'll look like that. All right? And there's no segments at this point. Okay? But you'll see that there is an area that is different. Right? This egg is made up of a bunch of cells, what we call a syncytium. The syncytium is a bunch of cells in one area or multiple nuclei, and they share cytoplasm. Okay? So this is what you end up like for a fruit fly. Adult fruit fly has a head, abdomen, thorax. Okay? The egg, okay, up top, developing, there's what we call follicle cells, there's a nucleus, this is the overall egg, right? Here's your unfertilized egg. Depleted nerve cells, this is what it looks like, your eggshell, your nucleus. Once it becomes fertilized, it begins developing, right? But the cytoplasmic determinants within the egg, the unfertilized egg, will determine which is front, which is back, okay? And so then you end up with a segmented embryo. You'll see the body segments begin. You have a couple, I think there's like 13 segments. Part of it being the head, part being the thorax, the rest being the abdomen. And they're color-coded to show you where they go, where they end up. So in the adult, where are the wings? The wings are attached to what part of the body? The thorax. In the adult, where are the antennae? On the head. In the adult, where are the legs? on the thorax. All right? So that's the normal order of body parts. Okay? But now if we change that, if we alter what's there, we can change what happens. You'll see. All right? So the eggs develop in the ovarian cells after fertilization, results in 3 stages. Okay, the segmented larva, basically 3 parts. And then the larva forms a cocoon in which it becomes an adult fly. Don't really care that much about the Drosophila. Okay? What I am talking about is the pattern. How do we know what's the head, the thorax, and the abdomen? Where do those segments start? Where do they finish? So where does the head end and the thorax begin? Where does the thorax end and the abdomen begin? All right? How do we know which side is going to be the head versus the tail? All right? And we're going to talk about these things called homeotic genes. All right? They control pattern formation in the embryo and in the adult stages. So that genes that are expressed, a certain set of genes are expressed at the head, another set at the thorax, and a final set or a different set and they are conserved over time. What do you see here? Wild type means the normal on the left. Mutant means something is different on the right. What is different about these 2 photos? What's on Amy Jo? The mutant has legs on the head. Is that the normal part? You think that's no, that's not normal. Okay? The difference being is that what has happened is that the homeotic genes that are normally expressed in the abdomen have been moved up. So the end of the head is no longer the end. What is normally the end of the head has now been changed to be actually the beginning of the abdomen. So therefore, during development, it recognizes that this should be where the legs go. And that's abnormal. All right, so the Nullstein, Vollard, and Wyckoff studied segment formation. They created mutants. All right, so disruptions in a certain set of homeotic genes, which determine segment 1, 2, 3, 4 to 13. Okay, and a lot of those mutations were embryonic lethal, meaning that they didn't develop into normal or didn't develop to adults. And Joe, you had a question?
So like in that situation, would the mutant probably die?
No. They might not die, just might not fly very well, have sensory powers or sensory disruption, but it might not be dead.
So the mutant might like pass on the mutation?
It could, but for this, if we were to continue it on, it could pass it on, but we're not. We're just looking at what the function of this gene is. So they weren't going to divide, or they weren't going to go on and reproduce. Okay? So overall, they found that there are 120 genes essential for normal segmentation, 1 through 13. All right? And the parts being head, parts being tail. Okay? Within those genes, they separated them into maternal effect genes and polarity genes. And so we're going to go from the 120 genes that are important for segment formation, and within those, we had certain cytoplasmic determinants that were derived from the mom during egg formation that were put into the egg. Okay? And they're already set up there, which started the program, which ran the program of early development prior to the cells being able to do anything on their own. All right? And this initially established the axis of the body of Drosophila. Okay? These are also called egg polarity genes because it gave a polar nature to it. It gave a distinct sidedness. It said that this was the front, this is the back. Okay? And ultimately, they also have a distinct effect on the. Okay? One of these maternal effect genes is bicoid. Okay? Bicoid is a morphogen. Okay? A morphogen meaning is that it's a transcription factor, and depending upon its concentration, it's going to elicit a certain response. So lots of bicoid means one thing, a middle amount of bicoid means another, low amount of bicoid means a third. Okay? Bicoid is the signal that determines the front half or the front of the body. Okay? So if I said lots of bicoid, that means that's the front or the anterior position. Okay? And this is all determined by mom. So mom, when she's producing eggs, the eggs are produced with a gradient of bicoid. All right? So here's the egg. Oh, we're not moving around. Here's an egg. Okay? And so red would be bicoid. Lots of bicoid here, a little bit less, and then none. All right? So if we did it in dots, whatever. Okay? So lots of bicoid towards the front. That's the anterior part. Very little bicoid means the tail. And somewhere in between there is going to be the middle. Right? An embryo whose mother has no functional bicoid, so we could shut it off in the fly. Right? The embryo, since mom is not affecting it anymore, there's no more front half of the body. It actually loses its function. And it's actually due in two sections. So here is your larva, normal, head, 1, 2, 3, right? Three thorax segments, and then 8 anterior segments, or abdomen segments, and you end up with the tail. Okay, you have 13 segments altogether. Well, now when you have that mutant, there's 2 tails. And the segments they identified the segments accordingly. It's kind of cool. The segments accordingly based upon genetic expression, and you say that 8, 7, 6, 7, 8, repeat of the segments. So this showed that bicoid, or the fact that a egg has an amount of bicoid at the anterior or in it, would determine whether or not there's an anterior portion to it. Okay? And because there is no bicoid in this, then the mutant, you end up with 2 posterior segments, which would not be condevelopment beyond that. All right? So this suggests that the product of the mother's bicoid gene is concentrated at the future anterior end and is required to set up the anterior end of the fly. All right? And so this is a gradient. All right? So if I did, say, front, or well, actually, let's do it scientific, anterior to posterior, okay, of the larva, bicoid would look something like this. Lots here, right? Something like that. Bicoid level. All right? High here, low there. Low at the anterior, high at the anterior, low at the posterior. Determining this is the front, the rest is the back. Right? Right? You get it? Okay, good. So, the bicoid mRNA, which is produced by the mom and stored in the egg, right, is concentrated at the anterior end of the embryo. After it's fertilized, the mRNA is translated, okay, into bicoid protein, and it diffuses from the anterior end, where there's lots of bicoid mRNA. Right? The result is your gradient. High at the anterior end, low at the posterior. So what happens if I inject bicoid mRNA into the posterior end of it? So that I have, what happens if I had high bicoid here and high bicoid here? And so this would be anterior, posterior. What's going to happen? You're going to end up with 2 heads. It's going to definitively say that wherever bicoid is high is going to be anterior. You're going to develop the head. You're going to have two head. That's the answer I forgot to. But this is what it looks like. All right, so here's your bicoid mRNA in a mature unfertilized egg. So this is a process called in situ hybridization, looking at where the mRNA is and staining for it. In blue, okay, you have lots of mRNA at the anterior end, and then on the right-hand side, you're looking at the protein. Okay, lots of protein at the anterior end, and then less as it moves from anterior to posterior. So that this was determined to be the front or the anterior axis, the rest is the posterior. So it gives a axis for formation from front to back. And in between there is the thorax. Now, I'm using bicoid as an example. Okay? Do you think there are others? Yes. So if I had one that determines the front, I probably have another that determines the back. Because when I got rid of bicoid, what did I end up with? I had two back sides, right? And that's exactly the case. You end up with another morphogen that is the posterior designation, and so on. So you have two morphogens that one determines anterior, one determines posterior, and then depending upon the levels of those two in between, you can the larva develops into the different segments. All right, high bicoid here, low, whatever is the anterior. Then as it moves down, so somewhere in the middle would be about equal between the two morphogens. Okay? What does this do? What does this mean? Why is it important? Shows that there's a determination of axis formation, right? That a protein, right, is required for some early steps of development, and it comes from the mom, right? It underscores the importance of mom's role in producing eggs, and that they actually are determining early development before fertilization actually occurs. All right, and the gradient principle, right, top to bottom, front to back, right, is determined by polarity and the position of the embryo is developed. Questions on bicoid and its anterior designation? No? All right, so on Wednesday, we will continue chapter 16. I think I will actually finish chapter 16 on Wednesday. Then we can get into viruses and then eventually evolution. I think I'll be able to finish chapter 16, 17, and 17 before spring break.
Our next exam is in April, right?
Yep. So think about all the stuff that's going to be on that.
I was going to say, it's over a
Whew.
month away.
That's going to be a lot. I might have to get in a different room for that one, so we can give you guys extended time.
Yeah. Good one. Give us more.
Do you think I'm playing? I'm dead. I'm serious.